Very good. It's great. Great to join you. Great to join you. I'm sorry, only remotely, but I hope it works. And I would like to give you a brief presentation on what fascinates me, and that is evolution, and also to think about how evolution works together with Christianity. And maybe we can also have some discussion afterwards. So I'm trying to share the screen now so you can see my presentation. And I hope this works. Can you see the slides? Very good. So first slide, God and evolution. And this is the topic of my talk, God and evolution. And here is a slide that I use to start almost every class that I give every year in Harvard. This is the first slide that I show to the students. Here are a few dates that are truly remarkable and truly important. And this is when it all happened. So you can ask the physicists, when did the universe come into existence? And the answer is 13.7 billion years ago, the so-called Big Bang. You can also ask, when did our own solar system come into existence? And again, the answer that physicists give is precise. 4.567 billion years is when our sun was born. Soon afterwards, Earth came into existence, and soon afterwards, Moon. Then we go into the biological dates. So the question is, when was the origin of life on Earth? And this we do not know for sure, but most people place it at around 4 billion years. The first date that is sort of coming with some certainty is actually bacterial life. By 3.5 billion years, bacteria were alive on Earth as they are alive now. And then it took a and the so-called eukarya are the higher cells. They came into existence 1.8 billion years ago. And complex multicellularity arose 600 million years ago. So only 600 million years ago, for the first time, you actually have structures on Earth that you could see without a microscope. So you could see life without the aid of a microscope only from about 600 million years ago. Then you can ask, evolutionary, what is the most relevant thing that happened in the last 600 million years? And really, by far, the most relevant step is the emergence of human language, approximately 1 million years ago, because human language gives rise to a new mode of evolution, not just genetic evolution, but now also cultural evolution. And this really makes us totally different to all the other animals on Earth. Then evolution is the theory that grows the tree of life. And broadly speaking, this tree has three very, very big branches. On one, bacteria, then archaea, they are also simple cells, and then the eukaryotes. At the very tip of the eukaryote branch, you see animals. And in this tip would be the tree that also leads to humans. So if we are asked to believe that evolution gives rise to all of this, we have to ask the question, what is evolution? And before we answer the question, what is evolution, we ask the question, what is it that evolves? And this was precisely explained by the evolutionary biologist Ernst Mayr, for example, who said that the carrier of the evolutionary process are populations. So the one thing that evolves are populations of reproducing individuals. And they can be cells or animals or plants. But populations carry the evolutionary process. Then mutation means that after some time, there is a new type emerging in a population. So something has changed now. For example, the genome has changed, or a new idea has emerged. And we would describe this as a mutation. And then selection, natural selection, means that over time, one type grows faster than another type. And this is natural selection. And for a long time, mutation and selection were considered to be the only ingredients of the evolutionary process. And this is kind of a bleak view, because natural selection, of course, is competition. And this is a fierce fight, everybody against everybody else. But in the last few decades, people have realized that selection is not the only aspect of evolution. There's something else that is truly important. This is cooperation. And cooperation means that individuals not only fight against each other, they not only compete, but they could also help each other. They could work together. And what we realize is that cooperation is a kind of master architect of the evolutionary process. So cooperation means there's a donor, the donor pays a cost, and the recipient gets a benefit. So we can think of a human person helping another human person, or we can think a cell that releases some valuable substance to help another cell. So here are 3 examples of cooperation that have fascinated me ever since. On the very left, we can see a kind of cooperation as it was already in existence 3.5 billion years ago. These are bacteria. Bacteria form filaments, and every so often a cell dies to feed the others with nitrogen. So here a bacterium cell gives its life in order to feed the others. And this was present already 3.5 billion years ago. Here's a kind of cooperation that came into existence 125 million years ago. These are social insects. These are workers that raise the offspring of another individual, the queen. Already Darwin noticed, why does this even exist? You know, how can natural selection lead to a perfectly designed individual that does not reproduce itself, but help another individual reproduce? And here's an example from our own time, only about 2000 years ago. And this is Vincent van Gogh's famous painting of the Good Samaritan. Of course, people help each other. And here the Good Samaritan helps another person. The question is, why do people help each other if it is just natural selection? And in this book, Super Cooperator, I describe 5 mechanisms for the evolution of cooperation. I describe how natural selection can favor working together over competition. And these mechanisms are called direct reciprocity, indirect reciprocity, spatial selection, group selection, and kin selection. I point out maybe the first 2. Direct reciprocity means our most important interactions are repeated. And in the context of a repeated interaction, cooperation can be a kind of winning strategy. Indirect reciprocity is most of our interactions occur in the context of reputation, where reputation matters. And when reputation matters, normally cooperation is a winning strategy. It is fascinating to observe that cooperation is the master architect of the evolutionary process. Cooperation is involved whenever we have something truly new emerging in evolution. Cooperation is required for the emergence of the first cell, the first bacteria, the first higher cell, eukarya, complex multicellularity, insect societies, and people. So therefore, I have called cooperation the master architect of the evolutionary process. This is also really radically changing the view of Darwinian evolution, because it is not only fierce competition, it is not only natural selection, it is also cooperation. So a question that often emerges in evolutionary biology, especially in the discussion with religion, is whether there's a tension between evolution and Christianity. And I also participated here in a program at Harvard with the Divinity School where we really discussed this question. And one tension that comes about, for example, is the view of creationism. But creationism is not what I want to discuss today, but instead, the biggest challenge that I see and that I experience in everyday life is actually scientific atheism. And scientific atheism is a kind of view that is prevalent in Europe and in the US among people, and where the statement would be that science offers all that is needed for understanding the world. Science suggests there is no God. Science shows that religious concepts are false. And the answer to this scientific atheism, in my opinion, and that is an answer that should be given broadly because people are often very confused, is the following: that scientists are actually often shallow in the God concept that they reject. So the God concept of Christianity is much more sophisticated than what is rejected by scientific atheists. I heard a professor, Peter Kreeft of Boston College, recently say very nicely, whenever I talk to a smart atheist, after about 5 minutes, I agree with him. I agree with him that I also do not believe in the God that he rejects, but it's not the God that we are talking about. The second reply to scientific atheism is really that scientific atheism itself is not science. This has to be clearly understood. It's not a scientific interpretation. It's not an interpretation of scientific theories. It's itself a metaphysical position. Scientific atheism for me is almost like an attempt to construct a religion of atheism. it is not science. It goes beyond the scientific interpretation. So the God, I think, that you would encounter in the major world religions is much more sophisticated than what is rejected by atheistic scientists. And here the beautiful quotes of Moses Maimonides, 1135 to 1204, God is a fathomless ocean of being, from whom all things originate. Beside him there is no second, beyond him there is no other. Or in the classical Christian theology, God, according to Augustine, is not an object in the universe. It's not caught in the flow of time, created the world ex nihilo. And these are very deep philosophical concepts. And science does not really negate them in any way. And further on, God is a creator and a sustainer of the world. So not only a creator who sets things into motion in the beginning, but who is needed to keep the moment into existence. And also that without which there would be nothing at all. And I think this very much relates to the teachings of Thomas Aquinas. And of course, you are the experts on this. So in my opinion, evolutionary theory describes some laws of nature which God instantiates to unfold life. And this is the proper understanding of evolution. Without God, there would be no evolution. There would be no universe. And this is the God that we are talking about in classical Christian theology. And also I try to explain to people that evolution poses as little a problem for religion as gravity. So whenever religious people have a problem with the theory of evolution, I would ask them, do you also have a problem with the theory of gravity? And most people don't have a problem with the theory of gravity. However, Newton, when he first understood the mathematical workings of gravity, he did have a problem. And Newton made the famous remark here, hypothesis non fingo. Even though I understand now mathematically what gravitation is, I do not make a hypothesis as to why there is gravitation. This is only up to God. So if we think of God and evolution, and if we firmly believe in the atemporal God outside the flow of time, it somehow seems necessary to actually hold the view that God lifts that entire trajectory of the world into existence. So instead of God setting things in the motion in the beginning and then lets it unfold according to some interplay of chance and necessity, the atemporal God, in my opinion, lifts the entire trajectory of the world into existence because has to will every individual moment into existence. And for me, this is the interpretation of Einstein's famous quote, God does not play dice. And so some of those ideas are described in the book together with Sarah Coakley, Evolution, Games and God, The Principle of Cooperation, published by Harvard University Press.
I want to talk briefly today about a couple of things. The CRISPR-Cas9 system and gene drive. Can you put these up on the screen? I apologize. Normally I speak with slides the same way Dr. Novak does, but Manuel told me that he wasn't going to let me do that today. So now, CRISPR-Cas9 and drive, I'm putting up here gene drives just to show you the phrases so that if you're interested in this, you can look up further information on the net later on. And can you give me one more? Okay, well, we'll work without the slide. Oh, okay. I'm showing you this because I want to show you that CRISPR is not an actual word itself, but just an acronym. And I'm not going to repeat all the different things. I have to put it up on the slide like this so that I remember it. And I hasten to assure you there'll be no quiz at the end of this on these things. So what is CRISPR and what is gene drive? So about 25 years ago, scientists found in single-celled organisms, bacteria, a kind of, if you will, an immune system to protect them from bacteriophages, viruses which actually invade bacteria. When viruses invade a bacterium, they inject their DNA through the cell wall into the bacterium. That DNA takes over the cell's bacterium and causes production of more virus particles. Well, it turns out this mysterious thing, which was not called CRISPR until, I believe, about 2005, when it encounters viral DNA, it cuts these viral DNA strands up, thereby protecting the bacterium. So in 2012, Dr. Jennifer Doudna, whose lab is actually just down the road at the University of California, Berkeley, found that she could use this same system to actually purify and alter DNA outside a bacterium. So what is this system? Well, it consists of two things. First of all, a nuclease, or an enzyme that cuts biologically, and what's called a guide RNA that takes these molecular scissors and guides them to the place where a cut is needed. Well, it turns out that you can design the system so that you can take a cell and use the molecular scissors, if you will, to cut out a piece of DNA, and with the guide RNA provides a template for the cell itself to replicate the part of the guide RNA and produce DNA. So, in other words, you can get into a cell and you can actually alter the DNA of the cell. Now, what's this being used for? Well, it's actually, so first of all, let me step back and say scientists have known it's possible to alter DNA for a long period of time, but it was very difficult. This use of this particular technique is actually very quick, and the cuts made and the replication of the new DNA is actually very precise. And so it's assumed a number of really key roles very quickly. First of all, it enables the construction of what are called knockout mice in weeks or months instead of years. Knockout mice are important in cancer research and other research because what they involve is an animal, mostly a mouse, where a particular gene's function has been knocked out so that it gives a very precise, if you will, animal model in which you can investigate the particular course of a disease, for instance, cancer, in the absence of a particular gene. It's also found a use in what is called personal medicine, and that is that you can extract cancer cells from individuals with cancer and use this CRISPR system to actually take each mutation in the DNA that characterizes the malignancy and test what the function of this mutation is so that you can try and pick out, if you will, the Achilles heel of the cancer so that we can move towards real personalized cancer treatment based on the individual mutations within a person's individual cancer. Okay, gene drives. What are gene drives? Well, a gene drive is basically a technique for increasing the prevalence of a particular characteristic or gene in a population. And in a gene drive like this, with the CRISPR technique, what you incorporate is into, and I would emphasize that gene drives work only in sexually reproducing species. So let's use an example of mosquitoes and malaria. What the CRISPR system enables you to do, actually, with say a species of mosquito which carries, say, malaria, is to modify the germline DNA of the mosquito, a few thousand of these mosquitoes, by incorporating into the DNA both these molecular scissors and, say, a gene which confers resistance to malaria on this mosquito. Now, you recall that in sexually reproducing species, every cell has two strands of DNA, and in the offspring, when mosquitoes mate, the offspring get one strand from each of the male and the female. So if you've altered either the male or the female using this CRISPR gene drive, what happens is, because you've incorporated all this material into that DNA, it picks up that the DNA from the wild-type mosquito requires the clipping and modification of the gene to the same pattern as your modified mosquito. And so what happens is that as progressively your core of mosquitoes, your, if you will, offspring of the modified mosquitoes increases, eventually this drive, this incorporation of the particular mutation within the mosquitoes drives its way through the whole population. And if you could, can you move the next? So in normal Mendelian inheritance, okay, with no gene drive or anything, the maximum you would expect to see over several generations of mosquitoes would be 50-50. Okay, 50% of the offspring will have a particular gene of interest, 50% won't. With gene drive, if you incorporate it early on, with each coupling, it transmits to all the offspring the modified gene. So then in the end, you end up with a type of mosquito which no longer can carry the malaria parasite. So other attempts or other ways that you might use a system like this would be to insert something in the germline DNA, say, of a mosquito with female that causes them to lay only infertile or sterile eggs, which would result in the extinction of that mosquito species. So we're left with something, and I would emphasize that gene drives with mosquitoes are in the process of being tried in the laboratory. Scientists have got together and determined that to the present time, none of the CRISPR modifications should ever be used in humans, and the gene drives should be experimented with only in the lab. Now, the thing is this. We have a technology here which actually could, at least in theory and probably in practice, eliminate a huge extent of human suffering. There are something like 400,000 to 500,000 deaths from malaria a year. all around the world, and over 2 million new infections, and many of the deaths occur in very young children under the age of 5. On the other hand, there are all sorts of concerns about a gene drive itself. What happens if the mutation that you're trying to incorporate gets incorporated at the wrong place? Because the guide RNA in CRISPR only requires about 18 or 19 of the bases to match up. So, in theory, it could actually cause mutations in other parts of a mosquito. Now, the other issues are that if you choose to try and eliminate a mosquito species, what effect does that have on the environment? What are the downstream effects of this? What's the downstream effect of the loss of biodiversity, in other words? And also there are potential biosecurity issues. This is not like constructing an atomic bomb. This work can be done with competent graduate students for a few $1,000s in a minimal laboratory setting. So there are potential real biosecurity issues here. Now, I mentioned that both gene drives and the CRISPR system are largely related or confined to the lab at the present time. However, there are strong currents wishing to use this for greater good. But with the biosecurity and other issues, we've got a conundrum here. I mean, everybody acknowledges that current national and international regulations regarding handling of biologics are inadequate for this. Who should control this? How should it be controlled? How do we incorporate into use of this the people who are going to be directly affected in this? I mean, if you're a North American, you might say none of this should be used at all, because we shouldn't be interfering with the environment in this way. But if you're an African woman whose child has just died of malaria and you're pregnant with another child, you're going to have an entirely different viewpoint. So there's a lot of disputing about this at the present time. And we recognize that popes from John 23rd, Paul VI, John Paul II, all express concerns in their encyclicals and letters about our lack of realization of the interconnectedness of species. And Pope Francis, as previous folks have said, have noted in Laudato Si, Our Common Home, that we really need to take care of not only the environment, but every creature in it. In fact, I mean, it was funny as I was reading Laudato Si, he was kind of channeling, Francis was channeling St. Francis, and St. Francis's notion that each and every creature is united to him by bonds of affection. And I was thinking to himself, he couldn't have been thinking that surrounded by a cloud of mosquitoes. But anyway, the issue, the problem here is that already these techniques are leading to the problem of pressure groups building up on each side. And pressure groups, as we know, don't talk to each other. They talk within themselves. What is really needed here is really open, constructive dialogue, which I've heard over and over again today, and which is historically a strong point of the Dominicans. So to bring the whole thing to a conclusion, I want to stop there and make apologies to any card-carrying geneticists in the audience. I'm an epidemiologist, and what do I know about genetics? And sort of close with finally by perhaps paraphrasing my friend Ron Austin's phrase, what are we going to do with this scientific whammo? Thank you.
Thank you. Our third presenter is Professor Robert John Russell, or Bob, as he's known here at the GTU. Bob is the founder and director of the Center for Theology and the Natural Sciences, and he's the Ian G. Barber Professor of Theology and Science at the Graduate Theological Union here. He's also someone to whom I'm especially indebted for his friendship and his academic expertise that have been an enormous help to me in my own work in science and theology.
Thank you, Michael, very much for that gracious introduction. And thank you all for being here and for inviting me to speak briefly to you today about a topic in physics and theology. And I'm going to read it because I want to keep it in my 10 minutes, so that'll guarantee closure. So, quantum physics and Christian theology, a short perspective on it. Quantum physics was developed over 3 decades, from 1900 to its completion in 1930. It is one of the 2 pillars of contemporary physics, the other being Albert Einstein's theory of relativity, published in 1905. Technically, quantum mechanics is only relevant at subatomic levels, and relativity only for enormous velocities far beyond those of everyday life. This means we rarely experience their direct effects in nature, and ordinary language is unequipped to describe them. Nevertheless, quantum processes underlie and give rise to the general features of the world, from starlight and sunlight to the color of plants, animals, fabrics, and paints, from the solidity of a table like this to the cold touch of metal like this. Because of quantum mechanics, when you stand by a pool of water, you see light both reflecting off its surface and refracting through its surface to the bottom of the pool and back. And why does one photon go off the top and one go through? We don't know. It is the kind of chance that was spoken of earlier. But quantum processes also produce specific macroscopic effects in the physical world. One way is such bulk effects as superfluidity and superconductivity. Another way is the amplification of individual quantum events to specific effects in the world, from the blip of a Geiger counter detecting the spontaneous decay of a single radioactive atom, to the human eye detecting a single photon. A particularly striking example is in the production of genetic mutations, which we also heard about earlier today, in DNA, and of course their crucial role in making biological evolution over 3.85 billion years possible. And quantum mechanics makes a wealth of technologies possible, from transistors to laptop computers, the World Wide Web, smartphones and GPS, from lasers to fiber optics and DVD players. So in an important sense, quantum mechanics is the right description of physical properties, unlike the descriptions given by classical physics and encoded in our ordinary language. If we want to bring quantum mechanics into our theological discussions about God, creation, evolution, human nature, and so on, we first must interpret it philosophically. Here we have at least 2 fruitful options. One, ask specific philosophical questions about quantum mechanics and then incorporate them into theology, or embed quantum mechanics in a complex metaphysical system, which also includes theology. In both cases, the results are very fruitful, both for theology per se and for bridging the gap between theology and secular knowledge and culture. Several of us, including cosmologist George Ellis and Christian philosophers Nancy Murphy and Tom Tracy, have taken the first route. We adopt the almost universally accepted, except by Einstein, philosophical interpretation of quantum mechanics, namely the Copenhagen interpretation of Niels Bohr and Werner Heisenberg. According to the Copenhagen interpretation, the kind of chance that pervades quantum mechanical processes is not like the chance in the everyday world, such as the unexpected juxtaposition of two well-defined causal trajectories. We call them car wrecks or roulette wheels. Instead, quantum mechanics is evidence of the complete lack of a sufficient physical cause in subatomic processes. And when we make measurements on these processes, the resulting quantum events, colloquially called the collapse of the wave function, are best understood as a transition, to use Aristotle's language, from a potential state to a single actual state. So you go from many potential states to the actualization of one. But no one knows why that one. That's the chance part. Using this interpretation and being clear that there are 7 or 8 competing ones that are very strong too, we can then construct a theological argument or account of special divine action, reflecting at least one meaning of our daily prayer, "Thy will be done on earth." Here in this account, God brings about scientifically unpredictable results while acting without intervening in nature and without breaking natural laws, if there be some. Getting on your version of Hume. We apply this to the Darwinian theory of the evolution of species, and it allows us to think about God acting in, with, under, and through DNA mutations over billions of years to bring about the diversity of life we see today. It provides one particularly robust form of a general approach to a theological view of evolution, which many call theistic evolution. This view includes such diverse thinkers as Pope John Paul II, Ian Barber, Teilhard de Chardin, Dennis Edwards, John Hott, Elizabeth Johnson, Erna McMillan, Jürgen Moltmann, Wolfhard Pannenberg, Arthur Peacock, Ted Peters, and Karl Rahner. Theistic evolution is enormously important for the viability, credibility, and intelligibility of the Christian message in today's culture, so driven by science. It is also tremendously important because it directly challenges the atheistic interpretation of evolution by such voices as Richard Dawkins. And at the same time, it renders superfluous such conservative Christian attacks on evolution as intelligent design. Others of us, most notably Father Michael Dodds, have taken the second route. In his profound work, Unlocking Divine Action, Contemporary Science, and Thomas Aquinas, Michael shows how quantum mechanics, cosmology, chaos theory, and biology have contributed to a wider understanding of causality in nature than was available in the classical world of Newtonian science. According to Michael, this wider understanding is highly compatible with Thomistic philosophy and its complex view of causality. The result is a deepened account of divine action in nature that brings together Christian theology and the natural sciences through the lens of Thomistic philosophy in very creative and promising ways. Michael also challenges the scientism, the pernicious view of atheists, again like Richard Dawkins, that uses science to support atheism. Michael thus offers both the church and secular culture a rich appreciation of Christian theology in responsible dialogue with science, and in doing so, he demonstrates the virtues of a Thomistic approach to the dialogue. So in conclusion, we are enriched today with 2 complementary and mutually supporting approaches to our theological engagement with science. Together, these strengthen theology's fruitfulness in articulating our shared Christian faith and increasing the church's capacity to powerfully engage and proclaim its good news to contemporary secular culture. Thank you.